only, 1 minute only. I'm starting YouTube and then
Yes,
Nikita,
sir.
you
Yes,
can start.
sir. Yes, sir. Yes, sir. Yes, sir. Should I start now?
No, 1 minute, 1 minute.
Okay, okay, sir.
1 minute only.
Yes, sir. Yes.
Yes, start now.
Sir, I think Dr. Anish, he has left his this meeting.
He has joined again.
Okay, okay, sir. Okay, okay. So good afternoon, everyone. So I'm Dr. Nitika Thakur and I'm working as assistant professor in the Department of Biotechnology, Shulin University, Himachal Pradesh. On behalf of Microbiologist Society India, I welcome you all to the Sunday Talkies, Researchers Talking from Abroad series. And today we have a very important speaker with us. Before we move on to our speaker of the session today, I would now request our president, Professor Arvind Deshmukh, sir, to kindly share the mission and vision of our society. Over to you, sir.
Thank you, Nikita ma'am. Really, I'm very happy. The scientist from Pakistan is going to present on our platform. It is a proud moment for us. Pakistan is our neighboring country, and we must have scientific interactions with scientists from neighboring countries. Today is the beginning. In the future, we'll increase our interactions more. Let me tell him, Microbiologist Society is a dominant association in India working in microbiology. We have 900 life members spread over India and other countries. So, sir, I'm very happy to welcome you on our platform, and I request Nikita ma'am also, you can introduce the guest, and I have made you host as well as Anish sir as a co-host. He can share the screen. Thank you. Thank you.
Thank you so much, sir, for sharing the mission and vision of Microbiologist Society India. So today's speaker is Dr. Anish Ali Shah. He's a HEC approved supervisor and currently working as assistant professor in the Department of Botany.
Nikita ma'am, wait.
Yes, sir.
Anish sir has again left. Wait for 1 or 2 minutes.
Okay, sir, sure.
He has left because of some technical problem, it seems. He will join. And it is the beauty of our lecture series that we are recording it, uploading on the YouTube, and making it available for about 20,000 students and teachers. Thank you. Thank you. Proceed, Nikita ma'am.
Thank you so much, sir. So I welcome today's speaker for Sunday Talkies, Dr. Anish Ali Shah. He's a HEC approved supervisor and currently working as assistant professor in the Department of Botany, Division of Science and Technology, University of Education, Lahore, Pakistan. His research field portrays his powerful interest in plant stress physiology and phytochemistry. He has done his master's in botany from Government College, Faisalabad, his MPhil from Government College, Lahore, and his doctoral degree from University of Punjab, Lahore. He adds to his name many research publications with an overall impact of 366.87. In addition, he has completed an international project in collaboration with Bulgaria, represented by Associate Professor Velkova. Furthermore, he serves as editor in national as well as international journals of repute. He adds to his name many prestigious awards and distinctions. In addition, he is serving as an active role in various types of academic councils, Board of Studies. He holds a tremendous research collaboration globally and serves the community and society through various initiatives as alumni member of African Association Rural Development Organization, ARDO, and member South African Association of Botanists. We are glad to have you today for our Sunday Talkies session, sir. And today, sir will be talking and delivering a talk on survival of plant under changing climatic conditions. So over to you, sir, for your relevant talk.
Are you listening? Are you listening, ma'am?
Yes, yes,
Can
sir.
you hear me?
Yeah, it is visible also, sir. Your PPT is visible.
Thank you so much, Dr. Nitika Thakur, for actually allowing me to present my research work and this topic on this prestigious society forum, Microbiological Society of India. I'm very thankful to the president of Microbiological Society of India to actually allow me to present my my topic here. So the topic that I want to present is survival of plants under changing environmental conditions. Can I start, ma'am?
Yes, sir. Sure. You can proceed with your presentation.
Yeah. The topic that we are going to present and we are going to discuss actually is a very emerging topic, and that is very much important with respect to changing environmental conditions. So we'll discuss the survival of plants, how we can make the plants survive under these adverse climatic conditions. So these are the contents of my lecture. So first we'll discuss what is the climate change, and then we'll discuss the effects of climate changes on the growth of plants. And then we will discuss some of the stresses in plants and multi-stresses in plants, and we'll focus on the general defense approaches in plants against stresses. Then we'll discuss about multi-tolerance. Ultimately, last we'll discuss about the mitigation strategies in plants. So first we'll discuss about the climate. You know, climate is the long-term weather pattern in a region, and that is typically averaged over 30 years. So this is a long-term weather pattern in a region. In the broad sense, climate is the state of the component of climate system, including the atmosphere, hydrosphere, cryosphere, lithosphere, and biosphere, and the interactions between them. So it is a huge term, it is a broad term, and it is a long-term weather pattern in a region. So we'll discuss how the climate change takes place with the passage of time. Since 1970, the global average temperature has been rising at a rate of 1.7 degrees centigrade per century, compared to a long-term decline over the past 7,000 years at a baseline rate of 0.01 degrees centigrade per century. So nowadays, temperature has been rising a lot. The rise in temperature is more as compared to the rise in temperature in the past. Temperature is very much rising. That is the main problem that we are going to discuss today. So these global level rates of human derived changes for exceeds the rate of changes derived by geophysical or biosphere forces that have altered the Earth system trajectory in the past. So nowadays, why these temperatures have increased due to various anthropogenic activities, various human-driven changes, various human-induced activities, and that's why the temperature, global average temperature nowadays in modern time, that has increased a lot as compared to the temperature rise in the past. So this is a graph that shows that the comparison between the rise in global temperature. We can see the global surface temperature has increased a lot due to human and natural drivers. You can see we can compare here the natural drivers only, and the other one that we are comparing is human and natural drivers. So these human and natural drivers have increased the temperature of the global surface a lot as compared to the natural drivers only. So humans have increased a lot. Humans have increased the temperature of the Earth's surface, and ultimately they have added to the Earth's overall temperature, global surface temperature, as compared to the natural drivers. So this is the temperature changes over the last 50 years. You can see that they have been changing over the last 50 years, and they have been increasing with the passage of time, and they continue to increase with the passage of time. So this is the change in surface temperature from present that might be predicted in future. If that increase in temperature persists in the Earth's surface, we might have more increase in temperature. Ultimately, our Earth will be facing heat at some point. So these climatic changes in a broader sense also includes previous long-term changes to the Earth climate. The current rise in global average temperature is more rapid than the previous changes and is primarily caused by human burning fossil fuels. So nowadays we say that the temperature is rising, the temperature of the Earth's surface is rising. That is mainly due to burning of fossil fuels. And these burning of fossil fuels are actually due to the human-induced activities, due to anthropogenic activities. So due to these climatic changes, deserts are expanding, and while heat waves and wildfires are becoming more common. Now, next we discuss about the climatic change impact on the food system. Now, the environment outcome of climate change, you can see there is increased salinization. There is increase in its salinity to the soil. Ultimately, plants are affected due to salinity stress. There is erratic rainfall. There is increased storm intensity. There is increase in the concentration of carbon dioxide in the atmosphere. There is increase in the temperature. There is increase in desertification or the formation of deserts. And there is also increase in the drought. Drought means deficiency of water. And these all are resulting in rising seas and increasing of floods. So these all are the environmental outcomes of climate change. And there is increased disease severity due to these impacts. And ultimately, the production or yield of crops is decreased. Nutritional content or nutritional value of the crops that is decreased. So now what are the physiochemical responses in plants to these climatic changes? There are various physiochemical changes in plants due to these climatic changes. Plant response to these climatic changes. So in the face of persistent and worsening climatic change, it has been crucial to investigate how natural population and community respond to novel environment. Several clear patterns have emerged due to these climatic changes. For one, the distribution of many species have shifted to historically cooler regions in poleward and upslope directions. So the distribution of the plant species have been changed due to these climatic changes. And these shifts have involved local extinction. So those plants that were not able to survive in these high temperature and adverse climatic conditions, they are no more there on the earth, and population is contracted. Population contractions have occurred at the warmer range edges as well as range expansion into historically cooler regions at the poleward latitudes and upslope elevations. So the plants have shifted from the high temperature regions to low temperature regions, and some of the plants have been extinct. So this is how the plant diversity is affected. Ultimately, the growth, ultimately the production, productivity, yield of crops is affected, and that is leading to the threat to food security globally. So additionally, many species now emerge and reproduce significantly earlier in the year, which is likely a biological response to shortened winter, earlier onset of the growing season, and prolonged drought. So as a result of these climatic changes, there are various changes in the physiology and biochemical reactions of the plants, and they have changed their reproductive cycle. So global agriculture is dependent on a relatively small number of crop species which have been there to optimize productivity within a relatively narrow range of environmental conditions. So the productivity of the crops that is affected due to these climatic changes, most developed countries have had to embrace modern agricultural technologies in order to achieve food security for increasing population, as well as to support agribusiness and increase generation. Now you know that climate change is drastically affecting the productivity of crops. At the same time, the population of the world is increasing, and that is a huge problem. And the production and yield of the crops that is decreasing. So we need to increase the production and yield of crops to for a sustainable agriculture to support agribusiness for next upcoming generation. And there is a prediction on the global scale that we require a huge, significant amount of trade in agriculture sector to meet the increasing demand of growing population. So there are some mitigation strategies that have been developed to combat the effect of such extreme events, which are destined to become much more frequent and are required alongside the global diverse drivers of agricultural production. So the concept that recent development in plants has the potential to address the challenges facing agriculture and food production is widely accepted. There are now a range of strategies available for enhancing sustainable crop production and resilience to climate changes, including high thorough output, single nucleotide polymorphism, genotyping, genome selection, and trait mapping. So in this lecture, we focus on how the plant can be made resilient to these climatic changes, how plants survival can be assured that despite of the diverse climatic changes and the negative effects of the climatic changes. So these tools that we are discussing and we will discuss, they're essentially not only for in-depth understanding of trait variation, but these tools are also for transformative engineering required to accelerate plant breeding efforts. So the pyramid approach to introducing favorable alleles and genes combinations are required. So we'll discuss some of the changes at the molecular level that we require to actually enhance the production and productivity of crops that it has been suffering due to climatic changes. So multi-stresses, you know, plants, they are land plants, they are living inherited harsh environment ever since their emergence. And they're facing a lot of stresses like high temperature, deficiency or excess water, high salinity, heavy metal, ultraviolet radiations. These all stresses affect the plant growth, and these all stresses are interrelated with each other. So the plant is not actually facing one type of stress, and these stresses have been actually due to the climatic changes. These stresses have been aggravated. These stresses have been increased. Their effect has been increased due to climatic changes. Now, we are not saying that these stresses are not present in the past. They were present in the past, like salinity stress and a drought stress plant were facing. But nowadays, due to climatic changes, these stresses have been now very much severe due to these adverse climatic changes. And most plants now, they cannot survive sodium chloride concentration at C200 millimolar. So these all stresses are linked together. Like in the last line, you can see more seriously, the salinity stress is interlinked with drought. That is another global issue which can be aggravated by extreme temperature. Now the temperature is rising. That rise in temperature is increasing the drought stress. And when we have drought in our land, ultimately the plant will be affected by salinity stress. And when these all stresses will be there, the plant will not be able to uptake nutrients from the soil. And you know, plant uptakes nutrients from the soil through soil solution from the rhizosphere. So ultimately, nutritional deficiency will also occur, and plant will not be able to grow. And even if some of the plant is grown in that area, that will have not all the nutrients that are present that need to be present in this plant for food security and for food safety. So in the field, plants are routinely exposed to unpredictable combination of different stresses rather than a single one. There is not a single stress, various stresses, and they are interlinked with each other. And that is even worse in the context of climate change, soil salinization, and environmental pollution. So these climatic changes, soil salinization, environmental pollution, they are increasing the severity of these multiple stresses. And particularly, we are already discussed on the demand of population growth. It is getting imperative to equip crops with multi-stress tolerance. So unless we are not able to equip crops with multi-stress tolerance, we will not be able to achieve sustainable agriculture, will not be able to achieve the sustainable growth and productivity of crops. So the occurrence of heat extremes in the context of climate changes have accordingly received much attention. So the foremost the effect that causes the reduction in growth, that is due to heat stress, and that heat stress related to drought, then drought to salinity to nutrition deficiency, and these are stresses then affect the crop. So drought is loosely speaking characterized by deficiency in the supply of water. The deficit condition could be related to some defined normal condition, or it could be relative to some demand for normal water. So these are the general defense approaches in plants against abiotic stresses. We have discussed some of the abiotic stresses, started from the temperature, then we discuss about drought stress, then we discuss about salinity stress. So these all abiotic stresses are affecting the plants. Like these abiotic stresses, they result in the membrane injury, and they damage the reactive species. They causes increase in the production of reactive species. So they result in the RS damage. And these reactive species, these are very much damaging, and they damage the macromolecular structure within the plant cell. Like a cell wall or cell membrane is composed of proteins. So these reactive species will actually interact with the proteins, and they will damage the membrane structure as well as other structure within the plant cell. So, and these abiotic stresses also cause protein denaturation, and these also cause osmotic stress. So these are some of the effects that are highlighted in red here. Like these are some of the damaging effects that are caused by abiotic stresses in plants. Now, what plants have some defensive approaches against these abiotic stresses. Like plants produce unsaturated fatty acid. Plants have some RS scavengers. RS scavengers in the form of some antioxidant enzymes, like what you see APX. APX is ascorbate peroxidase. GST, glutathione S transferase. And there are various other antioxidant enzymes that act to reduce the effect of reactive species. And when plants have to face protein denaturation, plants have molecular chaperones. So molecular chaperone, they are set of the proteins. They work together and to reduce the toxic effect of abiotic stresses, like heat shock proteins. And in response to it, osmotic imbalance, the osmotic stress, plants have compatible solutes like choline, glycine, butane. So these are some of the general defense approaches in plants against abiotic stresses. And there are various other signaling molecules in plants that act together to relieve the plant from abiotic stresses. Like there are some there are some signaling molecules like citric acid, hydrogen peroxide, hydrogen sulfide, nitric oxide, polyamines, calcium. These are all of the signaling molecules that activate the protein kinase, and they also cause a change in the transcriptional factor, and ultimately they cause the alteration in the gene expression. So that alteration in gene expression can lead to the activation of these defensive approaches in plants. So how we can achieve multi-stress tolerance in plants? So those techniques, those methods that we have discussed that lead to the defense against stresses. So we can just overexpress those factors, like cuticle forming agents can be overexpressed. Unsaturated fatty acid, those all enzymes that are involved in the production of unsaturated fatty acid, they can be overexpressed. Unsaturated fatty acid, they are important for maintaining the membrane fluidity. We can also overexpress. We can also understand the mechanism that are involved in the overexpression of all those enzymes that are involved in the reactive species scavengers, that active reactive species scavengers. And there also we can also have the overexpression of heat shock proteins, and we can have overexpression of the solutes as well. So we can achieve multi-stress tolerance by overexpression of the normal defensive strategies that we have discussed in the previous slide. Just we have to activate those strategies. We have to increase those defensive pathways by overexpression of the enzymes or pathways that are involved in the tolerance to against abiotic stresses. So that will help us to achieve multi-stress tolerance. And there we also need to understand different signaling molecule like abscisic acid, hydrogen peroxide, and nitric oxide, calcium. These all are linked together and they positively reduce abiotic stresses, and they link together and they involve in actually positive regulation of abiotic stresses. There are some signaling molecule like phytochrome, they are involved in negative regulation. So we need to understand the interaction, the crosstalk between signaling molecules, as well as these signaling molecules are also attached with the hormones like growth promoting hormones within the plant. So these signaling molecule can be increased within the plant, and their interaction with the hormones, their interaction with other hormones, growth promoting hormones, can be increased to achieve multi-stress tolerance. So we have nowadays various genome editing techniques and that can help to manipulate and modify the genes. Like genome editing is a method to generate DNA modifications at precise genomic locations, and these modifications can result in knockout or knockdown of one or multiple genes. So we can manipulate knock down one or multiple genes by genome editing, like we are editing the genes for achieving our benefits and without the permanent insertion of any foreign DNA. This is a recent techniques that we need to incorporate, need to do for a sustainable agriculture. So these are some of the genome editing applications for abiotic stresses. Like there are some of the genomes have been edited by different scientists, and some of the references are given here. Method that have been used, CRISPR-Cas9, that is a recent technology for genome editing, and this has emerged as a recent technology and emerging technology among the scientists for genome editing, and we can edit the genome of our plant or a crop, and we can achieve a multi-stress tolerance. So this genome editing has been applied in various crops. So genome editing for a range of rice diseases have been proven remarkably effective. Like CRISPR-Cas9 was used to produce knockouts of these family genes, and ultimately they included sucrose transporter, and that can be exploited by the pathogens. So ultimately, we can actually make the plant resilient against biotic stress, biotic as well as abiotic stress, by genome editing. So these are various reports that we have that genome editing can actually help us to achieve multi-stress tolerance. Like in the case of leaf blight here, genome editing effectively reduced rice susceptibility to a pathogen that is anticipated to become more damaging in some regions of the world due to climate change. So ultimately climate change is linked with the abiotic and biotic stresses. And not only abiotic stresses have increased, biotic stresses have increased because different kind of pathogens have been evolved, and they have been evolving. They're continuously evolving with the passage of time. So we need to enhance those strategies that can be used to reduce the toxic effect of abiotic stresses, and that can be used to combat those biotic agents or pathogens that are damaging the toxic effect of the that are damaging the crops due to their toxic effect. Now, CRISPR-Cas9 mediated knockout of this gene, an ethylene responsive gene. This is ethylene responsive. So every gene we can target which one gene that we need to knock out, which one gene that we need to enhance the expression. So this is previously implicated in blast resistance, and that has significantly reduced ligand size in response to being infected with leaf blast with no concomitant alteration to agronomic traits. So this rice was affected with a different pathogen, and that we can actually improve the quality of this rice and by knockout gene, by mediating the knockout gene and through a CRISPR-Cas9 technique. This is another genome editing approach. So editing a eukaryotic elongation factor ELF4G in rice using CRISPR-Cas9 that yielded plants entirely resistant to rice changro virus. So this is the biotic stress, and these viruses have also increased with the passage. Their severity have increased. They have been modified with the passage of time. So as the viruses have increased, as the pathogens have increased, as the bacteria have increased, their strains have emerged, and they are damaging the crops. So we need to also increase the defense mechanism in plants. We also need to alter the defense mechanism approaches in plants against these biotic and abiotic stresses that have been increasing due to climatic changes. Then we are linking these with the climatic changes. So these all are due to the severity of these stresses have increased due to climatic changes. So edible plants infected with virus had no detectable viral protein and exhibited high yield relative to wild type. So additional efforts are to engineer broad scale resistance to multiple rice pathogens simultaneously are described below. So you can see below that this rice is being engineered by CRISPR-Cas9 technique. In barley, this technique has been proven to be very effective. CRISPR-Cas9 mediated genome editing of MORC1, a defense relatively gene previously identified in Arabidopsis thaliana, and that is simultaneously increased resistance to Plumeria graminis, and that is the causative agent of barley powdery mildew disease, Fusarium. So various fungi and bacteria, they are actually attacking our plants, and we can have the recent techniques as the plant pathogens have emerged, and we have these plant pathogens and abiotic stresses and biotic stresses. They would not be able to combat against these stresses if we use the traditional approaches. So we need to enhance the new approaches. We need to enhance the techniques that we use, and we need to enhance the gene editing techniques. We need to use the recent techniques that we in genes to reduce the toxic effect of abiotic stresses and to reduce the damaging effect of biotic stresses as well as in different crops. So likewise, there is editing of a single locus in tomato that conferred broad spectrum resistance mutation to this gene. So loss of function mutants in the Arabidopsis thaliana maintain the high level of salicylic acid. So CRISPR-Cas9 gene edited tomato lines are more resistance to this bacteria Pseudomonas and different species of Pseudomonas, Xanthomonas species. So we can use this technique here. The scientists have used, and they have proven that this technique has been emerged to be very effective against biotic stresses, against various bacterial attack as well. So this is the overall diagram that shows the adaptation in plants to the changing climate. The ecological effects. First, we'll discuss the ecological effect that the climate changes have been so severe, and then ultimately we can have we can actually genome-wide editing, genome-wide genotyping, GVS analysis, and ultimately we can have we can develop climate-resistant plants. So there's a series of humans that we need to do with the plants to have climate resilient plants. So nowadays, this strategy is very much common. These strategies are very much common. They are not only dealing with the single strategy like in the past. We were only focusing on the effect of some fertilizers, effect of some PGPRs, and we're only focusing on the metabolic level. Now there are multi-omic strategies in plants, and that have to be developed if we want to develop resilience within the plants against these biotic as well as abiotic stresses. As you know, biotic and abiotic stresses have increased. We also need to increase the resilience in plants against abiotic stresses and biotic stresses. We need to increase the resilience, and we need to make them tolerant against different stresses. Like these are different multi-omics strategies in plants. Like we can go to metabolic level, metabolomics, and we can change we can actually have understanding of metabolites and how those are working in response to different stresses. And this metabolic pathway profiling analysis, that is very much important in plants, and that is going to under stress conditions. We can also have some proteomics. We can also do analysis of proteins, oral proteins. We can do proteome search, protein expression, structure and functioning proteomics, protein-protein interaction, and that is post-translational modification. So we not need to understand all these proteomic level approaches in plants. And we also need to understand the transcriptomics as well, as that shows differentially expressed gene analysis. Pathways need to be understood, and network prediction can be done by using different techniques and softwares. Molecular docking can be done, single cell RNA sequencing can be done, and even we can also go through genomics, like genomic level or DNA level. There is genome wide association studies, pan-genomic study, present and absence variation, structure variation. So to develop resistance in plants against different stresses, we need to have multi-omic strategies, not only single omic. Like in the past, we need to understand metabolomic, proteomic, transcriptomic, genomic, ionomic, and various other omic strategies that can be related to each other to achieve resilience in plants against stresses. And understanding plant biology with multi-layered information, not only single-layered information. The single-layered information will not help in actually developing resistance in plants against multiple stresses that can be used for one type of stress. So we have to accelerate crop improvement. We need to understand all these pathways, and then we need to understand how these pathways work, and then we can facilitate marker assisted breeding, genetic engineering, and genome editing. And we need to use recent genome editing techniques, miRNA editing techniques, CRISPR-Cas9 technique, and the various other recent technique that we can use to edit the genomes. And after editing those genomes, and then we can actually have understanding of the plants at the omic level, like this. We can understand the metabolic, proteomics, transcriptomics, and genomic level, and then we can develop a resistant plant against stresses. We can develop a resilient plant against multiple stresses. Because if you are going to develop resistance in plants against one type of stress, that is not going to work, because now climatic changes have gone so much severe. Now traditional research is no more there. We have to develop resistant plants. We have to work at molecular level, not only molecular level, at physiological level, at biochemical level. We need to understand how those all work together, how those all are coordinated, and ultimately we can actually develop some of the resistant plants against these multiple stresses, and we can have a higher production, higher yield of those crops. So this is another microbiome engineering techniques have been emerged, like different microscopic organisms that are in the rhizosphere. They can be engineered through different techniques, like their viruses can be engineered, and there is engineering of the soil microbiome. So this is the soil microbiome, and this is the rhizospheric region, and that need to be engineered to enhance the productivity of crops, because these are the regions where plant roots are in direct connection with the soil. So microbiomes, whatever the microbiomes that are present in the soil, and they need to be engineered. So we are talking about the beneficial microbiomes. And if we are only giving chemical fertilizers to the crops, that will not work, because ultimately it is converted. It is going to increase the waste. It is going to increase the pollutants in the environment. So we're not doing good with the environment if we are just focusing on intensive chemical farming, and we're just providing fertilizer. We're just providing chemicals to the plants. So we need to work on the microscopic organisms, living organisms that can be used along with those chemicals to accelerate the growth and to increase the production and productivity of many crops. So these microbiomes can be engineered, and we can engineer these microbiomes to have a sustainable agriculture. And there are various techniques. Again, we have to use the recent techniques to engineer these microbiomes. Not only we're not focus on one type of microbiomes or the microbiomes that we have used in the past. We need to develop some different strategies to incorporate, to introduce beneficial soil microbiome, and also to understand the multi-omic strategies that the plant develop to have resistance. And then if any microbiome is very much useful and that can work with other plant growth regulator, and ultimately it develops resilience within the plant, so we can work with other plants as well. So that's why I say that microbiome engineering is very much useful, and because it's not going to harm the environment, and there are already a lot of microbiomes that are present in the soil. So we can we need to engineer those microbiomes, microscopic organisms within the soil. So this is emerging mitigation strategies with respect to microbiomes. Like a lot of work has been done with respect to plant growth, promoting rhizobacteria, and still there are a lot of rhizobacteria that need to be assessed, and how those can achieve, those can help us to achieve multi-stress tolerance. Like they have been used to in plants to mitigate one type of stress, and like how these PGPR can actually induce the production of indole acetic acid, auxins, growth promoting a hormone within the plant. So these are new strategies that we need to incorporate and need to understand how these can be linked with the understanding of multiomic strategies in plants. And there are some microscopic organisms within the soil, like algae, like algal microorganisms. We can also use those for remediation of toxic pollutant within the soil and remediation of the toxic pollutant in the environment. And this is a new strategy, and very few research is going on in this field. And I have also read some recent papers on micro remediation in very well reputed journals. And micro remediation is just the focusing of the in that we are just focusing on use of arbuscular mycorrhizal fungi, AM fungi, like they are the endomycorrhizae, and we can actually use those endomycorrhizae along with other plant growth regulator, and those can work together. And now you have to find out the possible strategies, possible combinations, how these and arbuscular mycorrhizal fungi, different strains of arbuscular mycorrhizal fungi can work with which plant growth regulator, and how these are beneficial in remediation of the toxic pollutants, and ultimately they can make the plant resilient to a particular type of stress or multi-stresses. And bacteria already discussed, and there are some viral assisted remediation also going on. So these are some of there are a lot of papers, but these are some of papers that I've shown here, in which there is combined effect of this PGPR and zinc oxide nanoparticles. Now this PGPR actually assisted the plant in uptake of zinc from the soil. So that's how we can actually formulate different strategies, how we can use plant growth promoting bacteria or microbiome in addition with the plant growth regulator, and how it can be used to reduce one type of stress or many type of stress within the plant. So this is another paper, and then which we have used Bacillus subtilis along with silicon, and that was used to that in that study we found that the silicon uptake was increased. These bacteria, these PGPR and microbes, they assist the plant in uptake of many nutrients. But if we are using with some good combinations, they can be used to alleviate various type of stresses that the plant is facing, and ultimately we can have a great production, and we can have a sustainable agriculture. We can increase the productivity of crops. So this is another article in which we discuss some of the microbes that can be used to treat hexavalent chromium, and that is very toxic. So this is another slide in which we actually treated the plants by using a multiomic strategies. Multiomic strategies, we actually worked at molecular level, at metabolomic level, at enzymatic level, and these were supplied growth regulators, and these supplied growth regulator, they work together and they enhance the survival of plants at metabolomic level, not on a single level. So we discussed molecular level, transcriptomic level, enzymatic level. Biotechnology can be involved in this. So this is how we can actually achieve resilient plants against different type of stresses. So here at the end, I will discuss some of the special issues that I am going to edit. And if any one of you is going to submit your work, you are most welcome to submit your valuable work. Like I am editing currently these are open special issues, and that is in South African Journal of Botany, and we are editing a special issue. I am leading that special issue as our leading guest editor, and the topic is novel studies on physiological, biochemical, and molecular aspect of antioxidant role in mitigation of plant stresses. So here we are also discussing about the multi-omic strategies, not only a single strategy, like we are discussing about physiological strategy, biochemical and molecular level strategies. So this is another special issue that is also open, and in this we are actually focusing on, we will receive papers with respect to nanoparticles. You know, different nanotechnology has emerged as a good technology, and that in agriculture and people all over the world are using this technology to increase the yield and productivity crops and to reduce the effect of different stresses that the plant face. So this is another good journal from Australia, functional plant biology, and this issue is also open. So your work related to these topics can be welcome. So at the last time, just to say that I am very thankful to all of you, particularly Dr. Nitika Thakur and Microbiological Society of India. And if you are going to collaborate with us, you are most welcome. Thank you so much.
Thank you so much, sir. It was a wonderful talk. And yes, you have highlighted the most important criteria we are facing today. We are facing stresses across globally, you can say, across the plant stresses due to the climatic variations which we are facing. So yes, it was an interesting topic and the need of the hour. So I think everybody enjoyed the topic, and you gave the beautiful insights to this topic. And yes, I'm very happy to share that with the collaboration with Dr. Anis Ali Shah, our paper has been accepted in functional and integrative genomics, and it is basically based on the microbiome for sustainable agriculture and environment. So many thanks to sir for collaborating with us. And if you want to collaborate with the research proposals, you can go ahead, and sir has given the full information, and we'll be sharing the PPT also with you. So now the house is open for discussion. Like, you can come forward with your queries. So I think Professor Swaranjit, sir, you can come up with your query, sir.
Yes, Dr. Patankar is also there, who is a
Yes,
national coordinator of Microbiology Society. Dr. Swarnajit Singh is here. So I welcome all these scientists to interact with the Pakistan scientists. Thank you.
Sir, can I ask one question?
Yes, Dr. Anil
Yes,
Yadav.
yes.
Yes, sir, please.
This are you going for
No,
commercialization?
1 minute. I will introduce Dr. Anil Yadav. Anil Yadav is working in Hyderabad in one vaccine industry. And he has developed a very innovative idea and innovative products in vaccine. So, Dr. Yadav, you can ask the question.
Thank you, sir. My question is there. Are you going to commercialize these things? Hello?
Dr. Anis.
Are you going?
Yeah, are you going to commercialize? But
whether you are going to commercialize your research. Dr. Anis.
Sir, commercialize, sir, I am unable to understand the word commercialize. Like,
okay.
I am already interacting with many forums. Commercialize, like what you are saying, please.
Okay, here I want to ask simple. This, what is your research paper? Are you published, and really are you want to work with any company with your ideas? Are you ready to work jointly?
I would love to work with anyone. I'm already working in collaboration with different scientists all over the world, from China, from America, from Australia, and I have many papers with some Indian scientists and they are working in King Saud University, Saudi Arabia, and from all people all over the world. Science is not, there is no limit to the science. There's no boundaries to science, and I would love to collaborate with anyone from you,
Okay.
from your side.
Any market size? Have you know regarding market size?
Yeah, we have actually supplied these strains to the local market here in Pakistan, and we are working with the local industries, different industries, and to increase the production and yield of crops. So I can also work with anyone abroad, and if you're interested, we can also work and collaborate with each other, sir.
Oh, my question is any market size, because suppose we will go with this product. So what is the size of market? How many crores size market in Pakistan, in India, as per your experience?
Yeah, this is a very simple techniques that we have described. So it is up to agricultural farmers that how you can actually imply these techniques. You can actually imply all over the India, and all over the, we can also implying in Pakistan. These are simple seed priming strategies, and we are just using these microbes. So these can be implied at any level, if it is just depending on the implication and employment level and what you are trying to do. But these are the cost effective strategies. The cost effective, there's not too much costly, and we can use at any level.
Okay, because one of my Pune friend, they have seed manufacturing factory. They may require this type of technology.
Yes, I will be there, sir. Sir, I will be there. If you actually require anything, just contact me through Dr. Nitika Thakur, and I will be there to assist you in any matter, in anything that I can actually assist anyone there.
Okay, okay. Thank you, doctor. Thank you, Dr. Anish.
Thank you, sir.
Thank you,
Thank
Dr.
you so
Deshmukh,
much.
sir.
Hello.
Thank you, Yadav sir.
Hello.
Thank you, madam.
We have our national coordinator, Professor Sanjeev sir here. So, sir, over to you.
Yeah. Good afternoon to all of you, and good afternoon to Dr. Anish Ali Shah. Your talk is very wonderful, and you had talked regarding many more things, right from proteomics, metabolomics, and the other type of the things, the CRISPR technology. The thing is that whether all this research, it is going to be translational research. That is very important. And Dr. Anil Yadav, probably he pointed out the same thing. Whether it is going to a translational thing, that is actually on the commercial basis, and have you studied the economics of the entire thing? So
Right.
that is the very important issue. So all the researches, they are going in the laboratories, but they are not translated to the farmer or that particular level in a mass. So that is very important. So how you are planning for that? That is very important question.
Sir, thank you so much for your question. Thank you so much for your nice question. In fact, so we first thing is that we need to develop these strategies, and that is the need of the hour to develop. We're not going to develop strategy against one type of stress. We need to develop resistance against plants against different type of stresses, and ultimately we need to develop different techniques. And different patents have been approved of my research, and different industries we are collaborating. And already, sir, I have pointed to you, sir, that these are cost effective, and these are very much beneficial microbes, and that can be used along with different plant growth regulators to have a resilience in plants. And these are very much cost effective, mean they're not of high costly, and the method we implied is not too much costly. So we can actually we can do at any level. That does depend on the government to organization to support us, and we can actually present our ideas. It's depend on the government and the local bodies, the NGOs, how they can support us. Here in Pakistan, there many are collaborating like different industries, and we have also patents of our research in the recognition of this valuable work. So we are doing whatever we can do, and it's up to the local bodies and government. And these are actually applicable also. I can trust you. These are applicable, translational, and everyone is doing. Wherever you see multiomic strategy, just put multiomic strategies, you will find many special issues, and there are many special issues of my friends, and they are using multiomic strategy. This is a recent approach. We're not only talking about metabolomic or physiological. We will talk about somewhat more, because the stresses have gone more so severe, and we need to increase the production. Despite of the fact that we are being agricultural country in Pakistan, we are importing wheat. So this is another factor, because floods are there here. Floods have drastically reduced the yield of crops, and drought stress. Almost 1/3 of Pakistan is affected by drought stress. So we need to develop recent technologies, as the technologies that we used in the past, they are not working anymore.
Okay, so that is actually we want that it must be a translational, and it must go to the farmer, and it must come up in a public. Unless until
Yeah.
it comes in the public, otherwise it will remain the work that is confined to the laboratory. That should not happen.
Yes, sir. Yes, sir. Thank you so much for a nice suggestion, and we're working together with some collaborations with all over the world, and we will do at the government and even our own level at university, and with different NGOs of the local areas, inshallah.
Yeah,
Thank
it
you
was
so much, sir. So, hello, sir.
hello.
You can go with your query.
Yes, it was a very interesting talk. I really enjoyed the talk very much, and we really have to control this climate change all over the world, in the polar regions, in the hot regions, deserts. We have to really control. So, according to you, what the governments should do to control the change in climate?
Yes, sir. Thank you so much for your nice question. No, sir. The only thing that I started with presentation with climate, and climate changes have gone severe with the passage of time, and they're just due to anthropogenic activities. And these anthropogenic activities are decreasing plant diversity. So by planting, by increasing plant diversity, we can actually make the environment more good. Apart from this, and apart from this, we can also work on different mitigation strategies, how we can control plants by in laboratories as well as in the field. So this is how we can achieve by planting more and more plants. So because plants can act as an environmental buffer. They can resist the changes in the environment, and they might be the stability in the environment if we were planting more and more plants. And apart from these plantation, we can actually do some work in the lab to increase resilience in plants.
Yes, that's good. Now, I was wondering about one thing. What about the hardy plants,
Yes, sir.
which are
Yes.
very strong, like cactus, okay, water hyacinth, which is a menace? Are they also affected by the climate change?
Uh, yes, sir. You are right. All of these are affected to some extent, but those plants that are very much actually very resilient, like those plants that are drought tolerant, we can actually use the genes, and we can actually link those genes and understand the at genetic level in drought susceptible plants. And mostly these crops are affected, and the global food security is a huge problem in these developing countries. And Pakistan is a developing country, and most of the soil is very much suffering from drought stress, and we are working on drought stress, and there is salinity is also linked with that stress. So mostly our crops are affected. We are actually focusing on crops, agronomic crops, like wheat, rice, and some of the vegetables are being focused, but we are focusing on the crops so that food security cannot be compromised.
Right. Okay, thank you so much. Thank you.
Thank you so
Thank
much,
you
sir.
so much,
Thank you
sir.
so much.
So thank you, Anish, sir, for this wonderful talk. And now I would like to invite our national coordinator, Professor Sanjeev, sir, to share the vote of thanks.
Okay, good afternoon to all of you. And it's my privilege to give a vote of thanks for such a nicely organized and nicely given lecture by Dr. Anis Ali Shah, Assistant Professor, Department of Botany, University of Education, Lahore, Pakistan. Sir, we are very much thankful to you for giving a time to give and deliver the lecture and the webinar in our series, that is the researchers speaking from abroad. And I am also thankful to our national president for giving this opportunity, and also thankful to all participants, those who attended this particular lecture. Thankful to Dr. Nitika Thakur for organizing and anchoring the whole webinar, and she is constantly doing efforts for organizing this particular series. Thank you, Dr. Saranjit, and all other Dr. Anil for wonderful questions and other things, and also those who have given their opinion in the chat box. They are very nice suggestions and the chat that I have seen. So once again, thankful to you. Over to Nitika Thakur. Thank you.
Thank you so much, sir, for always encouraging us, and we'll be in communication with Dr. Anis for his future lectures too, and for the research collaborations. And I'm also collaborating with Dr. Anis, and it's a wonderful experience learning and sharing my views also, and learning from eminent researchers. So we don't only provide a platform only for like listening, but for implementations basically. So you can go ahead with the research collaboration, and we are providing you a global experience here. So you can collaborate according to your research interest, your research field, and you can connect globally. So thank you, everyone. Thank you, everyone, for joining in on Sunday Talkies, researchers talking from abroad. And we'll be providing you with the latest burning issues in these sessions. So stay tuned for the next lecture too. And thank you, everyone. Thank you, Dr. Anis, for delivering a wonderful talk. Thank you, everyone. So keep glowing and keep growing. Thank you, everyone. Bye-bye.
Bye-bye.
Thank you, everybody. Thank you.
Thank you. Bye-bye.